Hello, so this is the molecular biology portion of the e-report. So the first thing I'm going to be talking about is chromatin compaction. So DNA is the heritable material of life. It carries all of the information to make proteins that are needed for an organism to live, survive, and reproduce. DNA even contains within itself the ability to self-replicate through a process known as mitosis. So this DNA is within every cell's nucleus in any living organism. But for humans, the average size of the cell's nucleus is 5 to 8 microns. But all of this heritable material is nearly 6 feet long when stretched out in length. This doesn't seem to add up, but DNA has the ability to be tightly compacted into this tiny nucleus. Although DNA might be compacted and condensed just to fit into the cell's nucleus, the process to compact the DNA must be organized, regulated, monitored, and reversible. It needs to be reversible because there are genes on this DNA molecule right here. So all within this DNA, there are genes on it that have to be accessed at certain times for gene expression to occur. So we're going to have to get those genes if we need a certain expression to go on to keep the cell healthy and alive. For eukaryotic cells, like ourselves, we have chromosomes that just represent very long, compacted DNA molecules that are organized to be easily dealt out to daughter cells during mitosis or cell division. So right in the bottom here, this is a chromosome. DNA can be packaged in the same way rope is. In fact, the DNA is looped, coiled, spooled in an organized manner where the DNA is not tangled, but the individual genes are readily unpackaged and made accessible. The chromosome for replication, on the other hand, are more relaxed and less packaged so that the DNA is accessible for this process. And although they're relaxed and less packaged, they're still very organized. Since a chromosome is a long molecule of DNA wrapped up by proteins down here, the DNA together with proteins which wrap it are known as chromatin. So all throughout here we have chromatin going on. Right here is a chromatin. The basic unit of eukaryotic chromosomes are nucleosomes, and the fundamental components of nucleosomes are histone proteins. So right here it shows the nucleosome, and I'm going to show you a better image of these histone proteins. These histone proteins right here, these are all the histone proteins, are largely positively charged, which allows DNA to bind to these histones non-specifically because DNA is negatively charged due to the phosphate groups on its backbone. And there are 4 different histone proteins that are part of the nucleosome itself. So you have H2A, H2B, H3, and H4. And there's 2 copies of each of these 4 histone proteins, and they come together to form this octamer. And this octamer is just simply a protein complex made of 8 individual protein subunits. The unique thing about the octamer is that it is shaped of a spool, which allows 147 base pairs of DNA can be wrapped around this histone spool right here, which leads to our first level of chromatin compaction. Therefore, what we've done is decrease the overall length of DNA by spooling it. A nucleosome is considered when a histone octamer has DNA wrapped around it. So right here, because DNA is wrapped around this histone octamer, this is considered a nucleosome. So when the DNA exits one nucleosome, it leads to the next nucleosome, and there's this linker DNA composed of proteins. The pattern of linked nucleosomes is the essence of compaction, where 1/3 of the length of DNA is compacted by these nucleosomes. The least packaged form of DNA is known as the 30 nanometer fiber of chromatin. That is a coiled helix of nucleosomal DNA that compacts this DNA further. So in order to form this 30 nanometer fiber, though, it requires another histone protein, H1, which is right here. This H1 protein is known as the linker histone protein because it's positioned on the edge of the nucleosome, where the DNA exits the spool in a way to get to the next nucleosome. So interesting enough, though, H1 will change the path of the DNA to promote a more twisting structure, which favors the coiling of the 30 nanometer fiber. The 30 nanometer fiber is further looped and coiled to eventually form the 10,000-fold compacted mitotic chromosome. But it's still very poorly understood. As mentioned earlier, we need to access the DNA sequence at any time, and this is where the regulation of chromatin comes in. The cells regulate access to DNA and achieve localized unwrapping of needed sequences while keeping the rest of the chromosome tightly compacted. Cells use a variety of strategies to get the DNA sequence that are sequestering in chromatin, and each of these strategies requires an own set of proteins because it will require a cellular process. A common strategy is known as the chromatin remodeling complex. And right here, you see this is the chromatin remodeling complex, which uses the energy from ATP hydrolysis to simply move nucleosomes out of the way brute force wise. Chromatin remodeling complexes physically force DNA to unwrap from a nucleosome, and they simply thread adjacent DNA onto the nucleosomes. These complexes free up short regions of DNA, getting them out of the nucleosome and making them accessible for other DNA-related processes. These complexes also have the ability to rearrange nucleosome array, which are simply just clusters of nucleosomes that space the nucleosomes out more and expose the underlying DNA. The same way they do this, these complexes also can bunch up the nucleosomes and sequester the DNA again, making it a reversible process. These chromatin remodeling complexes are inactive during mitosis to this DNA tightly wound. So there's another strategy used for accessing DNA in the context of chromatin, involving the histone tails that project out of the nucleosome core. So that nucleosome I showed before, there's a tail that protrudes out, and this histone tail can be used to get DNA more accessible or tightened again. So these tails are rich in lysines, and they carry a positive charge on the side chains that interacts with negatively charged DNAs to lock nucleosomes in place. When we want to loosen this DNA, the lysines are acetylated by histone acetyltransferases, or HATs, that neutralize the positive charge, loosening the association with DNA, making DNA accessible, and weakens the nucleosome's stability. This is a reversible process, though, just like the chromatin remodeling complex, because acetylated lysines can be deacetylated by histone deacetylases, which are HDACs, which will restore this positive charge on the lysine and lock the nucleosome in place again, inhibiting the access to DNA. This regulation is crucial for compaction of chromatin to fit inside the nucleus and to allow accessibility to the tightly wound DNA at any given moment that the cell needs a sequence of DNA. So next, I'm going to talk about DNA replication. So DNA has evolved as the information storing molecule of life because it is a very stable molecule that can deal with harsh treatment and not be easily perturbed. This two-stranded molecule is tightly locked together by thousands of hydrogen bonds, holding bases together. If we wanted to separate the double-stranded bond, we'd have to boil it, because high heat would provide the needed energy to break the hydrogen bonds holding the two strands together. Sadly, cell doesn't have the ability to achieve these high temperatures to separate DNA for replication. Instead, there are specific distinct sequences of DNA known as the origins of replication that allow initiator proteins to bind at these origins of replication, pry these two strands apart by breaking the hydrogen bonds. And this is done because an individual hydrogen bond is so weak, but breaking a few bonds doesn't require a lot of energy either. The initiator protein causes a replication bubble where the site of DNA replication occurs. So this is seen here. This is the replication bubble that's forming. The site of active DNA replication is at the replication fork, a Y-shaped junction where the double-stranded DNA is separated and the new DNA strands are made using the old template strand. So if you see right here, this is the replication fork, and it's seen again over in this image over here. The replication fork will form on either side of the replication bubble and move away from the other in opposite directions. So the replication fork is able to move forward due to the DNA helicase binding to the replication fork. So we have right in this image right here, helicase right here, that's unwinding this DNA. DNA helicase is considered to be the leading protein in charge of replication because it is what separates the double-stranded DNA to allow replication to occur. So DNA helicase spins so fast and uses energy from the ATP molecules to pull this double-stranded molecule apart. Once DNA helicase passes through the replication fork, a protein known as single-stranded binding protein comes in and binds to the newly single-stranded DNA. And this is to keep it from reassociating with ' complementary strands. DNA polymerase cannot start synthesizing DNA from scratch because it can only link 5' ends of nucleotides to 3' ends of already there nucleotides. Thankfully, to save the day, RNA polymerase can start the replication process because RNA is so similar to DNA molecularly, and even better, RNA polymerase can make RNA strands complementary to DNA from scratch. So each newly synthesized strand of DNA is started with an RNA primer, made by RNA polymerase called primase. The RNA primase lays down the RNA primer, which is about a 10 base long primer, but it provides a free existing 3-hydroxyl end onto which DNA polymerase can bind and begin to add new DNA nucleotides. DNA polymerase is an enzyme that makes the new DNA strand using the old DNA strand as a template. DNA polymerase also makes a phosphodiester bond between 5' phosphates on an incoming nucleotide and a 3' hydroxyl on the end of the newly synthesized DNA strand. Their individual nucleotides enter the reaction sites of DNA polymerase as deoxynucleotide triphosphates. So this could be either DATP, DGTP, GCTP, or DTTP. All depends on what base you're using. And they carry the same energy as an ATP molecule. Therefore, the energy needed to power DNA replication comes from the cleavage of the DNTPs themselves, because when they cut 2 phosphates off of the DNTP, it releases a ton of energy. So, DNA polymerase alone would add a single nucleotide to newly synthesized DNA strands and then float away. And then eventually come back to add another base, but that's very inefficient. So to solve this inefficiency, we add a sliding clamp, which is shown here. So this sliding clamp protein will add to the clamp loader, and this clamp loader will be able to open up this sliding clamp so it holds on to DNA, and then it will hold on to and interact with DNA polymerase, and it does not allow DNA polymerase to float away, so that it can continuously add new bases to the DNA. So in order to finish up DNA replication, we have to take a minute and think about the replication fork again. DNA is always made 5' to 3', and new DNA is synthesized moving away from the replication fork from left to right. So the leading strand of DNA is synthesized continuously, while the lagging strand is made discontinuously, making the fork asymmetrical. The resultant DNA segments or fragments that make up the lagging strand are called Okazaki fragments, which will be joined together to make single-stranded DNA. The lagging strand will need more RNA primers due to these Okazaki fragments, and the leading strand will only have one RNA primer because it follows behind the replication fork. Nevertheless, we need to get rid of these hybrid molecules of DNA and RNA in both the leading and lagging strands, because this is not what we want. We want DNA, not DNA and RNA. So the only difference is that the leading strand will only have to do this once, because it only has one RNA primer, but the lagging strand will have to do this multiple times due to these Okazaki fragments, each having their own RNA primer. To get rid of these hybrid molecules, it requires 3 enzymes. The first enzyme will land on the 3' end of the fragment and chew up the RNA primer on the fragment in front of it. The second enzyme is known as a repair polymerase, and it puts down the new DNA in place of the just chewed up RNA. The third enzyme is the DNA ligase that will join the phosphate sugar backbones of adjacent DNA fragments to create one long DNA strand. And this is what completes the RNA replication process. So the next thing I'm going to talk about is DNA mismatch repair and the two mechanisms for repairing double-strand breaks. So thinking about it, mistakes are made by DNA replication proteins, or what we like to think of the machines that do occur in these DNA replications, but mistakes happen. And even worse is that there are natural and man-made caused carcinogens that damage DNA every single day. Any of these mutations, whether environmental or spontaneously induced, if left unrepaired, could potentially lead to catastrophic outcomes if they occur in vital protein coding regions. But like I know, cells are stupid. They have no way to determine if a particular DNA region is critically important versus another region. Therefore, the cell attempts to repair all mutations in real time because cells cannot tell which is a critical and non-critical gene region. They can't tell which one to fix and which one not to fix, that it won't matter. Luckily, there are mismatch repair systems of cells that are completely dedicated to fixing those mistakes that sneak by DNA polymerase proofreading. DNA polymerase makes one mistake in every 10 million base synthesides, but the error rate overall for gene copying is 1 in a billion. So mismatch repair systems fixes about 99% of the relatively few errors that DNA polymerase makes, and these errors are recognized by the distortion in the backbone of DNA. Normally, a mismatch is recognized in the newly synthesized DNA. That DNA is cut. The mispaired nucleotides and neighboring nucleotides are removed and replaced with the correct nucleotides and finally sealed in the DNA backbone. So this is shown right here in this process on this slide. If a cell has a mutated sequence, how does it know which base pair to change? The mismatch repair system must act fast and fix this mutation before the next replication. But the issue is still which base pair needs to be changed, or which one is the mutant. So, in bacteria cells, the cells use DNA methylation to mark the old correct DNA strand, so that when there's a mismatch, the cells fix the unmethylated or new strand to match the methylated or old strand because it's considered to be old and true. And this is shown on the left. Once the mismatch repair is complete, the new strand is methylated, marking it as old and true. But eukaryotes don't methylate old DNA strands. In fact, it's a big question mark. We don't know how new and old strands are distinguished in us, but it absolutely does occur. We do know the difference between old and new strands, but we just don't know how that process works. But it's not by methylation. The mismatch repair system is our absolute best defense against somatic mutations and even cancer. The most inherited predispositions to cancer are due to inherited mutations in the genetic coding regions that are part of this DNA mismatch repair system. So, for example, you inherit one mutant allele for a DNA mismatch repair protein from your mom and a wild type from your dad. So your cells are fine. You're good. You're living your best life. You have a mismatch repair system fully working. But then, later in life, you get hit and left and right with these lovely natural and man-made cause carcinogens, and that wild type from your dad is now mutated. And these mutations will rapidly accumulate in those dividing skin cells, passing on the mutation. One of these cells will pick up this secondary mutation, and you get cancer. Our DNA mismatch repair system is so critical to our defense against mutations and cancer. So any mutations within this mismatch repair systems, and we end up with cancer. So one of the worst things that can happen to your DNA molecules is double-stranded breaks. Double-stranded breaks in DNA is exactly what it sounds like. Completely severs a DNA molecule into two pieces. Double-stranded breaks can be caused by ionization radiation, mistakes at the replication fork, and free radicals. There's plenty more reasons, but those are the main reasons. These double-stranded breaks are potentially a life-ending mutation because the DNA is cut in half and there's no intact strand to serve as a guide for the repair. Thankfully, these cells have two mechanisms for repairing these double-stranded breaks. But one of these mechanisms is quick and dirty, which can lead to more complications such as mutations down the road. The quick and dirty mechanism for repairing double-strand breaks is non-homologous recombination, which is shown on the left here. It's a shorter process and it's a lot less efficient in the way that it can cause mutations down the road. Non-homologous recombination simply takes the two broken ends of the DNA molecule, files them down molecularly to make them blunt, and ligates them back together. So even though the double-strand break is repaired, yes, we got it repaired, it's all set to go, nucleotides are lost in the blunting process, and if these nucleotides were in this protein-coding region of genes, the gene will be inactive and you have a loss-of-function mutation. That protein and that region is just gone. It won't work. If this non-homologous recombination is done in a region that does not have any protein coding regions, this quick and dirty repair is normally fine for the cell. The better mechanism for repairing double-strand breaks, though, is homologous recombination, which is shown on the left here. But it will require a nearby DNA molecule that is extremely similar in sequence to the broken one. So although we have two complete DNA molecules that are nearly identical, these chromosomes aren't necessarily near each other in the nucleus, and if they aren't close by, this mechanism is not possible. In homologous recombination, the information on the nearby similar DNA molecule serves as the template for making the repair. So even if it isn't identical, but close enough in a pinch, it is viewed better than cell death and non-homologous recombination. Homologous recombination begins when a nuclease chews back one of the two strands of DNA even more at the double-stranded break. This free single-stranded broken end of the DNA then samples the nearby DNA molecule for similarity in sequence. If the sequences between the intact molecule and the broken molecule are sufficiently similar, strand invasion occurs. This creates a branch point where the two DNA strands, one from each molecule, cross the invading strand, and which is considered a D-loop, is then elongated by a process, by a repair polymerase. As the DNA is synthesized, the branch point moves forward just like the replication fork to allow additional DNA to be made with the use of helicase. Homologous recombination is completed by nicking the invading strands, allowing them to return to their normal positions, and sealing the nicks with DNA ligase. This leaves two completely intact DNA molecules. Homologous recombination is the most versatile repair strategy used to throw out mutated DNA. Homologous recombination even occurs during prophase 1 of meiosis, and it's responsible for crossing over. Meiotic cells intentionally cut their own chromosomes, creating these double-stranded breaks on purpose, and then repair those breaks using homologous recombination through crossing over. This results in DNA being swapped between maternal and paternal chromosomes and introduces a much higher level of genetic diversity in the chromosomes that we pass on to the next generation. So next I'm going to talk about the PIC, the assembly and the purpose of the PIC, or the pre-initiation complex. So in eukaryotic cells, transcription is a lot more complicated than in prokaryotic cells. So to start, there are three RNA polymerases known as RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. RNA polymerase 1 and 3 are utilized for RNA genes, and RNA polymerase 2 is used for protein coding genes. So we are going to be focusing on RNA polymerase 2. Sadly, our RNA polymerases cannot initiate transcription without some help from additional protein complexes. So the additional protein complex is composed of general transcription factors, or GTFs, that assemble at the promoter along with this RNA polymerase 2 to form this pre-initiation complex, or the PIC. Without the GTFs, transcription would be impossible because these general transcription factors form the PIC with RNA polymerase 2 to bring this RNA polymerase 2 to the promoter and release it from the promoter to initiate transcription. So there are 6 GTFs that are completely responsible for recruiting RNA polymerase 2 to the promoter, correctly positioning it, separate the double-stranded DNA of the gene, and launch RNA polymerase down the gene to transcribe. but I'm going to discuss the 3 main GTFs in detail of what they actually do within this PIC. The first GTF to recognize and bind to the promoter is transcription factor 2D, which includes a subunit on it called TATA binding protein, or TBP. This TBP will bind to the promoter specifically sequence known as the TATA box, which is rich in A's and T's. This is important because it is rich in A's and T's instead of C's and G's. So why? Because A's and T's only have 2 hydrogen bonds, which makes it easier to break them apart than C's and G's that have 3 hydrogen bonds. When when the TATA binding protein binds to the promoter, it kinks the DNA about 90 degrees, which makes this promoter unmistakable from other GTFs involved in this formation of the PIC. So it won't go to other promoters because it'll know by this 90-degree kink. Next, transcription factor 2A and 2B come in after recognizing this 90-degree kink DNA. And then after that, transcription factor 2F comes in and binds to the RNA polymerase II and aids the RNA polymerase II to the forming PIC on the promoter. Then transcription factor 2E will come and join the complex at the promoter. Like histone proteins, RNA polymerase has a tail at the end of its amino acid change that is accessible and modified. Transcription factor 2H comes in and phosphorylates this tail in specific places on the RNA polymerase II that signals RNA polymerase II's release from the promoter and allows for transcription to occur. Once the RNA polymerase II is successfully transcribing, the PIC complex or most of the GTFs will disassociate from the promoter, recruit an unphosphorylated RNA polymerase II to a promoter, and assist transcription initiation again. So this is all shown right here in this process right here. So the next thing I'm going to talk about is post-transcriptional mRNA processing. So even though mRNA has been transcribed, doesn't necessarily mean we're done. The next step for our mRNA is to go and be bound to ribosomes that are outside of the nucleus, but right now, the mRNA is still in the nucleus. Therefore, the mRNA must be exported to the cytoplasm for translation to occur. So eukaryotic cells have the ability for quality control where they inspect their mRNAs prior to the translation due to the compartmentalization. Our cells process the mRNA at the beginning and the ends to give this stamp of approval that the mRNA is complete and ready to move out of the nucleus into the cytoplasm and get translated. Only mRNAs with these additions on their beginning and ends are allowed to leave the nucleus. So let's talk about this process. First, the mRNA will receive a 5' cap, which is methylated guanine atypically added to the 5' end of our mRNA. So great, this marks the beginning of the mRNA as genuine and ready to leave the nucleus. So then the mRNA will receive a poly-A tail containing usually hundreds of A's along the 3' end. So this process is known as polyadenylation and requires multiple enzymes. After poly-A tail is added to the 3' end of the mRNA, the 3' end is marked as genuine. So once the beginning is capped and the end is polyadenylated, the proteins recruited to the 5' cap and the 3' poly-A tail will interact, resulting in good, mature, genuine mRNA to circularize. This tells the cell, we have a fully processed mRNA. It's good to go. But we still have to splice out the introns in the mRNA because they are just long, non-coding intervening sequence that are simply not needed. So on the left, we're talking about splicing these introns. So introns are considered remnants of extinct viral infections, magnets for mutations, and many other hypotheses that were made about introns' overall function. In order to splice these introns, we need a spliceosome that will scan for the limited conservative sequences at the ends and in the middle of the introns and ligates the exons, the protein encoding regions, back together. In this process, the 5' end is freed, and this end is attached to an A in the middle, which marks what is known as a lariat. The lariat saves the NTPs for other processes because NTPs would be expensive in the energy department to be remade. But there's also another method for splicing known as alternative splicing, which is shown on the right. And that allows the option to cut out exons if wanted to produce many proteins with different sequences, shapes, functions from a single gene. And this increases protein potential and modularizes protein synthesis. When RNA splicing is complete, the mRNA is fully processed and matured and is ready to leave the nucleus and enter the cytoplasm. So, translation. Once the mRNA was processed, matured, and left the nucleus, this mRNA is officially ready to be translated into proteins. Every 3 RNA nucleotides are known as a triplet or a codon, and these codons represent a single amino acid. So shown in the picture on the right, you can see the codon region on the mRNA. The correct open reading frame will start with the start codon, AUG, and end with the stop codon. Our cells have the ability to spot the correct reading frame by looking for a start codon. The codons in the mRNA do not directly bind to the corresponding amino acids, but need an adapter molecule to bridge the two. These adapter molecules are known as transfer RNAs, or tRNAs, that have 3 base anticodons on one end and the correct amino acid on the other end. So this is shown right up on top with the anticodon, and on the other end of this tRNA, there is the amino acid attachment site. This anticodon region directly base pairs with the codon using complementarity. The amino acid attached on the other end of the tRNA occurs in a process called charging. So enzymes that are responsible for charging the tRNAs with the amino acids are called amino acid tRNA synthesis. These enzymes are able to distinguish between tRNAs by the tRNAs specific sequences of nucleotides in the anticodon and the amino acid's attach point that varies between tRNAs. So without these enzymes, translation would not occur because these enzymes attach the proper amino acids to the tRNAs. The tRNAs serve as a nucleotide to amino acid dictionary and is the only cell that can understand both nucleotides and amino acid language. So to start translation, a specific tRNA known as the initiator tRNA will associate with the free ribosomal subunit, the small one, particularly. The eukaryotic initiating factors, the EIFs, will then bind to the 5' cap of the mRNA to the small ribosomal subunit. So this ribosomal unit will keep the mRNA engaged as it scans for an AUG start codon with a CAU anticodon. When the AUG is found, the initiator tRNA will base pair to it and the large ribosomal subunit joins the complex. So this larger ribosomal subunit has 3 slots that are each shaped perfectly for the tRNA. These 3 slots are the amino acid tRNA, which is the A site, the peptidyl tRNA, which is the P site, and the exit site, which is known as the E site. So the A site carries a single amino acid, the P site carries a peptide chain of amino acids, and the E site allows the peptide chain of amino acids to lead. So, once the large ribosomal unit joins the complex and associates the initiator tRNA carrying a methionine will join the P site. It is important to note that the initiator tRNA is the only tRNA that can bypass the A site and directly enter the ribosome P site. Since the initiator tRNA is in the P site, translation can begin to occur, and the next tRNA is placed into the A site. So the initiator tRNA will now move to the E site after that, and the tRNA that was in the A site will now move to the P site. So only 2 tRNAs are allowed in the ribosomal complex at a time. Translation will keep occurring until a stop codon lands into the A site. Once the stop codon is in the A site, proteins known as release factors, which is shown here, will enter the A site, bind to it, and catalyze the release of the polypeptide amino acid chain from the tRNA in the P site, freeing it from the ribosome. So this is shown right here on the right. We have these release factors going into the A site, and it's releasing the polypeptide amino acid chain. So after the release of this polypeptide amino acid chain, the two ribosomal subunits will dissociate and the mRNA is released, which is shown in the last portion on the right, labeled number 3. The ribosomal subunits in the mRNA have the ability to repeat this translation, and the newly made protein folds into its native 3D shape, does its job that it was made to do. And one of the best things about translation is that at any one time, a single mRNA is being translated by multiple ribosomes, each at different points of translation. It's known as a polysome, and it's able to boost the protein output rates. So this is good. We want to get as many proteins as we can, so this is happening at multiple ribosomes at different points of translation to get more proteins. So now I'm going to talk about the combinatorial control and mediator. So in eukaryotic cells, the genes are turned on or off by committees of proteins. So therefore, multiple proteins are needed to turn a eukaryotic gene on or off, and this allows eukaryotic cells to have the potential for genes to be responsive to multiple signals, and this is known as combinatorial control. So in our cells, there are multiple regulatory proteins that work together to carefully and precisely control the expression of a gene. So normally in our cells, a typical gene is controlled by dozens of regulatory proteins, both repressors and activators. So once all these regulators have transmitted their own individual signals, the end results of this gene is a committee decision. So by committee decision, it means that if there's more activators than repressors votes wanting the gene on, the gene is on. If more repressor than activator votes are wanted, the gene to be off, the gene is off. So this type of regulation is complex. It's a democracy vote, but allows fine-tuning of gene expression. So in eukaryotic cells, transcription factors bind to DNA and then loop out intervening sequences to reach the target gene promoter. Activators do not have the ability to recruit RNA polymerase II to the promoter, but they do aid in transcription initiation. So activators function more to grant RNA polymerase II permission to proceed with transcription and leave the promoter. So this is shown on the left and on the right. You can look at it at either aspects. Mediator is a huge protein complex that sits on the end of RNA polymerase II. And it's important because it basically is considered the relay complex. The mediator is the one who hears every own individual signal that these transcription factors, the activators and the repressors that regulate a gene, transmit. They transmit their answer to the mediator, and the mediator is the one who hears them out. The mediator can even receive signals from upstream regulators at enhancers. So this is shown on the right. You see these enhancer regions. So they can receive signals from upstream regulators at enhancers, or they can send activating or repressing signals. That's what the mediator can do. The mediator will then integrate all of the answers, finalize a single result, which means activate or repress that gene, and send the final answer to RNA polymerase II. So if the result was to activate the gene, the mediator will then have the RNA polymerase II leave the promoter and start transcribing that gene. If the result was to repress the gene, the RNA polymerase II will not leave the promoter to start transcription. This mediator is conserved throughout eukaryotes, and all of these eukaryote mediators have the same central core. The additional subunits attached to the central core of the mediator vary between different eukaryotes, but the mediator is absolutely a necessary component of transcribing genes, and it allows this fine-tuning of gene expression to occur when needed. The last topic I want to talk about is RNA interference, or RNAi. And this is a mechanism used by cells for gene silencing. But it has to, it is required to have a double-stranded RNA. What? I thought that was crazy when I first heard it. But yeah, you heard it right, double-stranded RNA. Once the RNAi machinery recognizes the double-stranded RNA and the single-stranded RNA with the same sequence, the RNA machinery destroys the double-stranded RNA and that single-stranded RNA with complementary sequence. So if a double-stranded molecule with your gene sequence is recognized by an RNAi, you will never make a protein from that gene again. That is guaranteed. Any mRNA that is transcribed from the gene will immediately be marked for immediate destruction. RNAi is an extremely potent response and is believed to have evolved as a defense against a viral infection because viruses are primarily the natural source for double-stranded mRNA. RNAi is triggered by a double-stranded RNA, shown on the right. You start with a double-stranded RNA. And this is fully base paired, longer than 21 to 23 base pairs long. And once this RNAi is triggered, an enzyme called dicer, shown on the right below the double-stranded RNA, will cut these long double-stranded RNA molecules into 21 to 23 base pair fragments. So these double-stranded fragments are called small interfering RNAs, or siRNAs, that are bound to the RNA-induced silencing complex, RISC. So this is shown right below dicer. You see the siRNAs, and then you see this RISC down here, and it is forming this complex of siRNAs and RISC complex. So RISC will use one of the two RNA strands from the siRNA to identify other RNA molecules that have complementary sequence. When these RNA molecules with complementary sequence are recognized, RISC will destroy these mRNAs. It is important to note that less than 50 siRNAs can silence 1,000 of these pre-existing mRNAs. And the reason for this is the amplification of siRNAs by an RNA-dependent RNA polymerase, the RDRP, which is seen on the left of this siRNA and RISC complex. So this shows that the amplification of siRNA, and that's why it's going back up from the siRNA mRNA complex back up to siRNA. So after RISC cuts an mRNA, there are two fragments left, and the RDRP uses one of these fragments as a template to generate double-stranded RNA without a primer. So this double-stranded RNA is cut by dicer to create siRNAs for RISC, and it keeps going on and on and on, causing amplification. So that's where we get a whole bunch of more siRNAs from these two fragments that were already cut by RISC, but RDRP decides to amplify this siRNA. But we lack RDRP, so we do not have the ability to amplify RNAi. We don't amplify. But RNAi is so unique that we can even exploit it in a laboratory setting, because RNAi can be utilized to prevent protein expression of targeted genes. RNAi offers a shutdown on command option, where it will express the siRNA molecules and achieve an instant stoppage of protein synthesis. So therefore, if it wasn't shocking then, it's definitely shocking now. RNAi is a pretty powerful mechanism that any double-stranded RNA or mRNA with completely complementary sequences have an already determined fate: death. And that wraps up all of the 8 topics for the molecular biology portion of the e-report. Thanks for listening.